Hello everyone, welcome to PathoMed. Today, let us discuss a topic of invasion and metastasis of tumor, which is also the metastatic cascade. Before jumping into the theory, let me first briefly summarize or tell you a synopsis kind of thing for better understanding. So, the first image it shows you a normal epithelial lining. The cells, the epithelial cells, these are the epithelial cells. They are resting on a surface called basement membrane, intact basement membrane. And these cells are tightly adhered to one another. They are stuck to one another with the help of these red color markings, which are called adhesion molecules, also E-cadherin, also called E-cadherin. It is an epithelial marker. It is an epithelial marker, epithelial adhesion molecule which binds these epithelial cells to one another. They stick to one another closely and they do not move away from one another. This is very important character feature of epithelial cell, normal epithelial cell. Whereas coming to a tumor cell, tumor, cancer cell, it can be due to any insult to the nucleus, like genetic damage because of mutation of suppressor genes, any like microbial insult or chemical and radiation insult, as we have discussed in our previous video of microbial and chemical radiation oncogenesis. Because of any insult, because of any alteration in the genes, the cell turns up to be a cancer cell. It loses its normal architecture, the size and shape. It becomes pleomorphic. See, normal cell, the nucleus is basally located. It's small in size. It maintains a certain size and shape. Whereas when you take up a cancer cell, nucleus is bigger and it becomes hyperchromatic. The nucleus to cytoplasmic ratio has changed when compared to its adjacent normal cells. Increased NC ratio. This is a cancer cell. So the cancer cell also you have observed there are no these adhesion molecules. I have not drawn these adhesion molecules. These adhesion molecules are not present. It is lost. So it loses this epithelial marker so that it gets detached from its adjacent neighboring friend normal cells and it can travel through the basement membrane, through the surrounding tissue, through the blood vessel to a distant organ. If a cancer has arisen in a liver, it wants to travel to brain. It wants a foreign trip. It has to travel to a distant place. So it detaches itself from the surrounding normal cells and it enters into the surrounding tissue and through it has to break that barrier. It enters the circulation and then reaches its final destination. In order to do so, the cancer cell has to detach from the neighboring cells. So it loses this adhesion molecule. It becomes a free bird. And now you see, it has become a free bird. And to invade, firstly, it has to break down this basement membrane. The basement membrane is breached. There is a breach in the basement membrane. And also, it has to enter into this extracellular matrix, the stroma. Stroma is present in between the cells, or the extracellular component is a stroma. It is present within a tissue. It has to enter this matrix. So it has to enter means it has to have a certain attraction for this ECM. To come and bind to ECM, it has to have some attraction points. The attraction points are it expresses this integrins. This pink color is the integrins. And also within the what is basement membrane made up of? Mainly made up of collagen, yes, and also laminin. Laminin, it is a glycoprotein helpful for migration. It has laminin and various other components which we'll discuss later. So, in order to attract this ECM, it expresses these integrins which help in attachment, also these laminin receptors which help in attachment of this tumor cell to ECM. And after it comes and sits in the ECM, it does not want to sit there permanently. It has to go somewhere else. So to break this barrier, it has to break down this ECM. It now breaks down the ECM by releasing what? The cancer cells or the surrounding stromal cells itself. It releases certain proteolytic enzymes so that this ECM is broken down. This ECM has to be broken down. So it is again set free to travel somewhere else and few of these enzymes are MMP9, UPA that we'll discuss in detail now in a few minutes. After breaking down this ECM, I mean the stroma, the interstitial tissue, it has venules, capillaries, lymphatic capillaries as well. It gains access to this blood vessel or the lymphatic channels and enters this blood vessel. If at all it enters a lymph node also, if at all it enters a lymph node also, there is communication between the lymphatic channels and also the arterial channels. This communication, and so this is how the tumor cells also gains access to the systemic circulation. It can enter into the systemic circulation as well. If at all it catches up direct blood vessel also, it enters the blood vessel. It forms a thrombus, and this thrombus breaks through this blood vessel and upon reaching this distant organ, for example, it wants to go and settle in the brain, or else in the brain also, in the liver or the brain also. After reaching its destination, final destination, it comes out of this blood vessel and sits in the final organ. This is a brief summary. And in this process, the cancer cell, which is basically an epithelial cell, in for its easiness to migrate through this blood vessel or the stroma, it changes itself into a mesenchymal cell. See all these blood vessels and this interstitial tissue, this is a mesenchymal tissue. So it transforms itself into a mesenchymal epithelial cell, transforming into a mesenchymal cell is called epithelial mesenchymal transition. That we'll discuss. This concept we'll discuss in detail now. This is a brief summary I have given. So firstly, let us start with the introduction. Invasion and metastasis, they are the hallmarks of malignant tumors. Benign tumors are confined, they do not metastasize or they do not invade, whereas the malignant tumors, they invade and metastasize. These are the characteristic features, hallmarks of malignant tumors. And for this invasion and metastasis, there are certain regulator genes, the metastatic signature genes, they promote metastasis or the invasion. Metastasis suppressor genes. Suppressor means they try to suppress this metastasis. The mutation of the suppressor genes favor metastasis, whereas the expression of the signature gene promote metastasis. Few examples we'll learn what are the signature genes. Few examples are adherin, which this is a signature gene whose expression it promotes metastasis in case of rhabdomyosarcoma. Rhabdomyosarcoma is a malignant tumor of skeletal muscle, particularly snail and twist. These two are important in breast cancer, and there's another gene MYR10B. And coming to the few examples of suppressor genes, mutation of suppressor gene promotes metastasis. Few examples are NM23. This is a kiss, you kiss the skin, so melanoma. So mutation of kiss causes melanoma, or the promote melanoma. Sorry, KAI1. It promotes the prostate cancer. These are the few examples. Firstly, the first event of invasion is the breach of basement membrane and stromal ECM. As I have said, the epithelial cells resting on a basement membrane, this is a cancer cell, this it has to breach through this. Breach of basement membrane is the first step and it enters the ECM. It also breaks down the ECM. This is the first step of invasion. Invasion is it moves to the local spread is invasion. Locally it breaks down the basement membrane, enters ECM, breaks down it. Breach of basement membrane and stromal ECM is the first ever step in invasion. After invading this interstitial tissue, that is the stroma, the tumor cells penetrate the lymphatic channels or the vascular channels. Now, coming to this concept of epithelial and mesenchymal transition, I have said this is useful for migration. To achieve this migratory behavior, the epithelial cells, that is the cancer cell, which was originally the epithelial cells, acquire properties close to mesenchymal cells. Why it has to travel through the blood vessel and it has to travel through the interstitial tissue, which are mesenchymal tissues. It be a Roman in Rome, so it transforms itself into mesenchymal tissue for its easy passage in this journey. This is called the epithelial mesenchymal transition, which favors metastasis. Normally, the epithelial cells form continuous sheets because I have said, see, this is a continuous sheets. Like it rests on a basement membrane, it forms a continuous sheets. It has certain polarity also, and they have strong intercellular connections. They're strongly, tightly sticking together to one another, tightly sticking together to one another. They're strong intercellular adhesive junctions. That is the E-cadherins. See, this is the E-cadherins, strongly bound to one another. Whereas the mesenchymal cells, they do not have the strong adhesions. They do not have strong adhesions, but they do form this loose network. The extracellular stroma it is very loose, fibrocollagenous stroma, very loose, myxoid stroma, as we have discussed in earlier histopathology slides. The stroma is very loose. These mesenchymal cells are loose network. They form loose network of ECM. So the epithelial cells become like mesenchymal cells. This is normal epithelium cells. They become mesenchymal, loosely dispersed within, so that they can easily pass through this mesenchymal tissues. This is the basic concept of epithelial mesenchymal transition to to achieve this migratory behavior. For this EMT, there are few genes which promote this EMT. Particular importance is given to snail and twist. Snail and twist, which we have studied as an example in breast cancer in the previous slide. These two genes they promote this EMT. Particularly the snail one, it down regulates E-cadherin. E-cadherin is an adhesion molecule, as we know. This is reduced. The cells adhesions are reduced. This one, it is broken down. I mean, these are reduced. So the now cells are set free, decrease cell adhesion. This is done by snail 1. Whereas snail 2, it activates the cytoskeleton. The cell's cytoplasm has cytoskeleton, like intermediate filaments or microfilaments, which are necessary for the cell movement. So it activates the cell's cytoskeleton, which is necessary for migration. The cell has to move through, right? For this migration, cytoskeleton has to be activated. For that, snail 2 is helpful. And in this EMT, the epithelial markers are downregulated. Yes, as I've said, E-cadherin is decreased. That is the meaning. Epithelial markers are downregulated. And obviously, mesenchymal markers are upregulated. It has to become like it acquires the properties of mesenchymal tissue, mesenchymal cells. That means mesenchymal markers are upregulated. What are these mesenchymal markers? Increase in vimentin, increase smooth muscle actin, which are necessary for movement, of the cell. And hence the cell acquires this pro-migratory, that means movement, pro-migratory phenotype. The cell is said to have acquired this pro-migratory phenotype. And in the recent studies, this drug called cystatin C, it is known or it has been studying for this uh, studies say that it inhibits EMT, it inhibits this transition. So studies have been made to prove its efficacy in breast cancer. Studies have been done to prove its efficacy in breast cancer by inhibiting EMT. This is statin C, recently in studies. Let us study the steps of metastasis one by one. Firstly, the detachment of tumor cells. Now, as we have said, they're tightly bound to one another. The neoplastic cell separates out. One of the cell, which is a neoplastic cell, it separates out. It has detached from the neighboring cells because of, as I said, E-cadherin is the culprit. Loss or reduced expression of E-cadherin. See, this is a normal tissue. The cells are tightly bound to one another. Cells are tightly bound. They're stuck to one another with the help of a glue. This glue is the E-cadherin glue. Whereas in cells turn out to be tumor, that is neoplastic cells, the glue is lost. The glue is lost. There's no glue over here. They are separate from one another. That is, they're detached from one another. Tumor cells are detached from one another. Next step, what is the first step? What is the next step after detachment? It has to attach to ECM. That means it has a breach, it has passed through the basement membrane. Breach is there, as I have explained earlier. Now it enters into the ECM. This is the ECM. ECM, it has to attach to the ECM. So ECM, it is made up of collagen, type 4 collagen, laminin, fibronectin and vitronectin. After detaching from one another, they enter this ECM. It has to attach to ECM. There are certain attraction molecules, as I said, they express integrins and also laminin receptors. This is a tumor cell. They express the integrins, for example. They express the integrins and also laminin receptors. For example, this is the laminin receptors. They express this so that it gets attached. It is attached to the ECM. It is attracted towards the ECM, pulled towards the ECM, and sits in the ECM, helping attachment of tumor cell to ECM. Now it is attached to the ECM. It is sitting within the ECM tumor cell. After sitting, it has to be ECM. It does not want to sit within the ECM permanently. It has to move through. It has to go to some other place. So this ECM now becomes a barrier. Now it has to degrade this ECM. After tumor cells have invaded the ECM has been broken down. For what? For migration. With the help of what? Proteolytic enzymes. Proteolytic enzymes break down the ECM, and these enzymes are released by this tumor cells itself and also by the surrounding stromal cells. Within the extracellular matrix are nothing but the stroma. Stroma has certain cells, mesenchymal cells, surrounding stromal cells and the tumor cells itself, they release proteolytic enzymes. Four important these enzymes are matrix metalloproteinase, that is famous to call MMPs, and urokinase type plasminogen activator UPA, and elastase and cathepsin. These four enzymes are released by the tumor cells and also by the surrounding stromal cells of the ECM. Particular importance is given to MMP9, matrix metalloproteinase 9, because it breaks down the type 4 collagen. ECM is made up of type 4 collagen also. One of the component of ECM is type 4 collagen. It is broken down. Hence, MMP is called the collagenous matrix cleavage. It has a role in collagenous matrix cleavage. Whereas the urokinase type PA, it has two kinds of functions. It activates the elastase and cathepsin. Once it is released, it in turn activates elastase and cathepsin, which cleave, that is break down non-collagenous matrix, non-collagen matrix like glucuronic acid, hyaluronic acid, sulfates, all this present within the matrix, these are broken down. Non-collagenous part is broken down. Also, it has an autocrine function. Autocrine means it is released, the cells which release it, the released product, it acts on the same cell and also the cells, neighboring cells, the adjacent cells. For example, this is a cell, it releases certain substance. If it is releasing a certain substance, this is a molecule, it acts on this cell and also on the adjacent cells also. It acts on this cell and also adjacent cells. The same cell producing the substance, the substance is acted upon by the same cell and also adjacent neighboring cells. That is autocrine function. Autocrine function. So the tumor cell releasing this UPA, this stimulates the tumor cells itself to synthesize the collagenase. Collagenase again breaks down the type 4 collagen. And see, we have discussing this ECM is broken down, that is, it is cleaved. The ECM, what are again these cleavage products? These waste products are also have a role. It is not of waste, it has some certain use. The cleavage products of the ECM also have use in what? Growth promoting, like by acting as a basic fibroblast growth factor. It is helpful for angiogenic activity also, like it acts as a vascular endothelial growth factor, and also it has certain chemotactic activities. It attracts platelets. So all these are necessary for in the process of this metastasis. These malignant cells, they become more motile after this invading that breaking down this ECM. It becomes more motile and they proliferate to become progeny of cells within the ECM. They become motile, proliferate, and reach this blood vessel or the lymphatic channel. The tumor cells enter the lymphatic and the vascular channels, where they form a tumor emboli. The progeny of cells, they form an emboli. Emboli means a clump of cells. And along with the clump of cells, clump of tumor cells, they are covered with the fibrin. This light blue is the fibrin, and also platelets. Purple dots are the platelets, and WBCs. All together form a tumor emboli. They're composed of tumor cells, fibrins, platelets, fibrin, platelets, leukocytes. All these form a tumor emboli within the vascular channel, lymphatic channel, or the blood vessel. After forming an emboli, it does not want to stay travel only or swim only in the blood vessel. It has to reach its destined or designated organ. So after reaching that is from liver, if at all it enters the circulation and travels to the brain, after reaching that specific organ, it has to come out of this vessel, the blood vessel. So firstly, the tumor embolus, it also expresses in the ECM. To attract the ECM, it, as I said, it expresses laminin receptors and integrins. That those two substances, they have role in ECM. Now the tumor cells also express certain molecule called CD44 molecule. This CD44 molecule do not have a role in ECM, but it has a role in the blood vessel. CD44 has a role in blood vessel. They express the tumor cell. It expresses CD44. It expresses CD44. CD44, it attracts the tumor cell towards the endothelium. It pulls. Now the tumor cell, here the tumor emboli is present. It is attracted towards the endothelium because of this CD44. This embolus attaches itself to the endothelium. This is the endothelium. This is the endothelial cells. Tumor embolus is attached to the endothelium. It is pulled towards the endothelium because of CD44. So after tumor cells, they attach to endothelium. Now the endothelium reacts. Someone has attached to itself, so it reacts now by retracting. It separates itself. The cells move away. See, there's a gap in between. Wherever the embolus is attached, there's a gap formation. There's a gap here. So retraction, that is means the endothelial cells move away. They give a way for the embolus to go out. It retracts. Reaction of the endothelium. Retraction. Now the tumor embolus, they escape this blood vessel. They escape the circulation. After coming out of the blood vessel also, they have to break down this. It has a basement membrane here. It has to break down this basement membrane. It totally comes out of the vascular compartment. Now it forms a metastatic tumor in its designated organ, final organ where it has to reach. So this is how the metastasis is complete. The metastatic cascade is complete in this way. Firstly, wherever the tumor cell is formed, it breaches the basement membrane. Firstly, it breaches the basement membrane. The basement membrane breach. Now it has entered the ECM. ECM could degrade. It broken down the ECM. Broken down ECM has been dissolved. From here it enters the blood vessel. From the blood vessel, tumor embolus is formed within the blood vessel. It attaches to the blood vessel. Embolus attaches to the blood vessel. It breaks through the blood vessel and settles in some destined organ. Settles in some destined organ. This is a metastatic cascade. And all this is happening, and the host immune system is not responding. So how it is escaping this immune system? Evasion means escaping the immune system. Tumor cells escape the immune system in immunocompetent, means healthy individuals, by what? Selective outgrowth of antigen negative variant of tumor cells. These tumor cells also express certain antigens, tumor antigens, which are recognized by the T cells. Cytotoxic T cells, they recognize these antigens, they come and attack the tumor cells and kill it. But this is not happening because the few of the tumor cells, they do not express these antigens. They do not have any antigens on the surface. This antigen negative variant of tumor cells, they survive. Selective, these are selected and these outgrow. This is selective outgrowth of antigen negative variant of tumor cells. This is one of the reason. And there is loss of MHC1 molecule. See, MHC1 molecule these tumor cells to CD8 T lymphocytes. These molecules are lost. Either this is one of the mechanism, or the other mechanism can be immunosuppression due to TGF beta PD-1 ligands, and galactines, which are secreted by tumor cells. Tumor cells produce these three components, three substances, which cause immunosuppression, and it can be tumor antigen. I mean, few of the cells expressing this antigen. For example, this blue color is a cell. These blue dots are the tumor antigens. They can be present, but they are masked by synthesis of glycocalyx. This purple thing is glycocalyx. These are non-immunogenic. Non-immunogenic. They do not evoke the immune response. These are non-immunogenic. When these numbers outnumber the antigens, the tumor cell is masked. They are not recognized by the T cells. So glycocalyx outnumber. They are not identified by T cells, and they are presently more in number. The tumor cells cannot be recognized by the T cells, so it escapes the immune system, immune response, and there can be an apoptosis of CD8 T cells. These are the various methods by which the tumor cells they escape the immune host immune response and survive. And anyways, in immunocompromised patients like in HIV/AIDS or those or in immunosuppressant therapies, already the T cell count is low. They have this weaker immune system. So